This lecture will be covering RNA metabolism, also known as transcription. And the title slide gives a pretty decent picture of how this process works. It looks a lot like DNA replication, which we talked about last time. The main difference is you're only using one of the strands to create a new strand of RNA. And so this RNA polymerase moves along the DNA, the double-stranded DNA, and it's unwinding and rewinding the DNA as it moves along. And so it doesn't unwind the entire length of DNA. It's just really this moving bubble, if you will. Okay, and then you're adding nucleotides, RNA nucleotides now instead of DNA nucleotides. You're adding to create an RNA strand. And we'll talk about some of the proteins involved in doing this. Right, so this is from chapter 26, and it covers RNA metabolism. Mainly what we're talking about is transcription, and that's what's also known as DNA-dependent synthesis of RNA. So we're using DNA as a template to synthesize RNA. Okay, we're also going to be talking about some of the ending parts of this, and that's the capping and splicing of the RNA, how it is processed, the messenger RNA, how that's processed. And then we'll be talking about some RNA-dependent RNA and DNA synthesis at the end of the lecture. But what's the most important is definitely the process of transcription, and then some of the processing that occurs to that. Right, so here's a problem just to keep in your head. Try to list, when you're going through this lecture, try to list the similarities and the differences between the DNA polymerase that is used in DNA replication and the RNA polymerase that's used in transcription. And a little bit later in this lecture, we'll try to list all of the things that come up. So an overview of RNA metabolism. RNA is synthesized in cells using DNA as a template, and that process is known as transcription. Transcription is tightly regulated. Okay, so you don't have every gene in the cell always being transcribed at a given time. So you regulate this process to control the concentration of each gene product or protein in the cell, so it's in an optimal level. You don't want to be wasting energy making proteins you don't need. Okay, being mainly single-stranded, RNA molecules can fold into very compact structures, very different than DNA, and they can also have very specific functions. And actually, some RNA molecules act as catalysts, and these RNA molecules that do act as catalysts, catalyze reactions, are known as ribozymes. So they're very similar to an enzyme, but they're not made out of amino acids. They're made out of nucleic acids, and specifically ribonucleic acids. We'll talk a little bit about those near the end of the lecture. All right. Most eukaryotic ribonucleic acids are going to be processed after they're synthesized, and some of those process ways that they're processed include the elimination of introns. Remember, we've talked about introns from chapter 24. Those are pieces in the gene that have DNA coding, but they don't lead to proteins. So those pieces of introns joining the exons are cleaved out. So you only have exons. You have other ways of processing. You have polyadenylation at the 3' end. So you add a string of A's at the 3' end, and then you cap the 5' end. Right, here we have a cartoony figure from the book of an RNA polymerase. And it's transcribing the DNA in this direction, from left to right. Right, the active site is shown here, and at the active site you have the 3' end of the green RNA strand that we're building. And this, we can just assume this is our messenger RNA, and we're trying to transcribe a gene here that's in the DNA. Right. There's 2 strands of DNA, right? There are 2 strands separated here in this what's called a transcription bubble. So it's very similar to the replication bubble, just much smaller. Okay. The non-template strand is up here, and the template strand is down here. And the template strand is the one that the RNA is being added to, and the RNA is a complement of that template strand. So for instance, if you have a G residue or nucleotide, there we go, G in the DNA, you'd have a C in the RNA that's being added to it, a complement to that. So it's adding to the template strand. Okay. And we'll mention this later, but the non-template strand, also known as the coding strand, you would get a very similar sequence of your RNA that's being built to that non-template or coding strand. Right. If you're interested, only about 17 base pairs of this is actually unwound at any given moment. Okay, so actually a pretty small area of that DNA. Right. And the 5' end would be the where the triphosphate would be. That's the first residue that was used in that strand. So if we talk about this transcription, you need a DNA-dependent RNA polymerase. Okay, so the DNA-dependent means you need that DNA as a template, and it's an RNA polymerase because it's building a chain of RNA. You also need ribonucleotide triphosphates. So you need, just like when we're trying to replicate DNA, in the DNA polymerase, you need those dinucleotide triphosphates. With ribonucleic acids, in the RNA polymerase, you need the ribonucleotide triphosphate versions, right? And the big change here is that in DNA you have the base T, but in RNA it uses the base U instead of T, okay, in place of that. So that's something to keep in mind. Right, and you also need magnesium. Magnesium's important in these, just as it is in DNA polymerase in the active site. Okay. It's the new chain of RNA is being synthesized in the 5' to 3' direction. Okay, so that's very similar to the DNA polymerase. When it makes a new chain, it also makes 5' to 3'. Okay. And so you have this reaction where you have your nucleotide chain, RNA nucleotide chain. You add a nucleotide triphosphate to that, and you get the chain is one longer, and you get pyrophosphate given off of that triphosphate, nucleotide triphosphate. Right, and when we're doing this, the template is being copied in the 3' to 5' direction. Right. No primer is required for this. Okay, that's one thing that's different than a DNA polymerase. No primer is required. And then the first 5' triphosphate, the first residue there, remains throughout transcription. In DNA, that is a monophosphate. Sort of a cartoony picture of this. RNA polymerase comes in. You open that double helix in that localized region, and then you can begin transcription, right? And it begins at what's known as the +1 site, right? And it moves down. As it moves down, it makes a chain of messenger RNA that is sort of spit out of the end of the RNA polymerase. If you want to see this in action, this is a good video. It'll be in the description if you want to look at that. Might help seeing everything in motion. So if we look at a double-stranded sequence of DNA, if we have a template strand, that's what our messenger RNA is going to be complementary to. So if we read that template strand in a 3' to 5' direction, that's going to be our 5' to 3' RNA transcript, and it's going to be complement to that. So, right, if we have a G on that template strand, it's going to be a C in the RNA. A C would be a G, and so forth. Okay, but remember, there are no T's in RNA, so every time an A is in the template strand, and we would need to use, usually you'd use a T there to be the complement, now we're using U's. Okay. You'll also notice that the RNA transcript is basically going to be the same thing as the non-template strand or coding strand. Okay, they're going to be both in the 5' to 3' direction. Sequences are going to be very similar, except T is going to be replaced with U in the RNA. Okay, both strands of our double-stranded DNA may encode for proteins. So it's not as simple as one of the strands is always going to be the coding strand and the other is always the template strand. It very often is the case that you can do this transcription in both directions, and so there are multiple gene products. Okay. In adenovirus, that's one of the things that cause the common cold. This happens. It has a very large linear genome, and each strand encodes a number of proteins. And you can see that, there's these green arrows indicate where a gene is on that DNA, and what would be RNA transcripts from that, and it goes in both directions. Okay, so if RNA polymerase transcribed the following strand of DNA, what are the first 5 bases of RNA that would be made? Okay, so remember, try to think about this. We have our non-template or coding strand and our template strand. So the RNA is going to be made to be complementary to the template strand, which will mean it looks very much like the coding strand. Okay, so our sequence, the first 5 bases here are going to be these first 5. Okay, so it would be A, U, C, C, U. All right, and remember, U is going to be used in our RNA instead of T. Right, so it's going to follow the same sequence as our non-template or coding strand, and that's because it's using the template strand as the pattern. Okay, so this would be our messenger RNA that would be created from these 2 double-stranded DNA if this was the template strand. Okay. Well, the movement of this RNA polymerase causes local supercoiling. So you can imagine that there's a small little bit of DNA that's being unwound, and as this motors along the DNA, you have to unwind the DNA in front of it, and by doing that, you're going to encounter this local supercoiling of it. And in particular, ahead of that polymerase, you're going to be overwound. Behind it, you're going to be underwound. And so you need topoisomerases to eliminate those supercoils. Right? As a reminder, topo 1 and topo 2 in prokaryotes, what they do, topo 1 relaxes DNA by removing negative supercoils. Topo 2 actually induces negative supercoils, which can relax a positive supercoil. Right? So topo 1 would act on the sort of behind the polymerase. Topo 2 would act in front of it in bacteria. Eukaryotic topoisomerase 2 relaxes either negative or positive, so that one could be acting both in front and behind the polymerase in eukaryotic cells. Right, and just in a picture form, right? If we're moving our polymerase along this DNA, kind of unzipping it in a local manner and then rezipping it, we're going to get negative supercoils behind and positive in front, and we need the topoisomerase to alleviate those. The active site of RNA polymerase is almost identical. You can think of it as almost identical to the active site of DNA polymerase, and we showed basically the same figure in chapter 25, right, where you have the 3' hydroxyl getting a nucleophilic attack to the alpha phosphate on the incoming nucleotide triphosphate. Okay, and this is all stabilized with magnesiums and aspartic acid residues in the active site, right? The only difference here is we're not a deoxyribonucleic acid. These are actually just ribonucleic acids because they still have their 2' OH groups, okay? But the mechanism of this is the same as DNA polymerase. Okay. This is just another figure showing that with some more colors in it, and maybe a little bit easier to look at, right? But again, it's showing that same mechanism, the 3' nucleophilic attack on the alpha phosphate of the incoming nucleotide triphosphate, and then you're losing the pyrophosphate. Bacterial RNA polymerase has a lot of subunits, and we'll go through some of these. 2 alpha subunits function in assembly and binding to the up elements, and we'll talk about what up elements are. It stands for upstream promoter. So that's what the alpha subunits do. There's a beta subunit, which is the main catalytic subunit, and a beta prime subunit, which is responsible for DNA binding. And then a sigma subunit directs enzyme to the promoter. We'll talk about those in a little, some detail, slight detail. And then the omega appears to protect the polymerase from denaturation. Okay, but the most important ones, I would guess, alphas, 2 alphas, beta, beta prime, and sigma. How they look in more of a real world picture, this crystal structure shown here, how they all fit together. Probably more likely you'll be looking at sort of a cartoon depiction of this. So it's sort of a tetramer here, and this would be the core of the RNA polymerase. You have the 2 alpha subunits, right? They're going to be binding the up elements. Beta prime is binding DNA, and then beta is the main catalytic subunit. Okay, to make the whole holoenzyme, you do need this sigma and omega subunits as well. But we're going to be primarily talking about what's happening here in the beta subunit when we talk about the actual polymerase in creating a chain of RNA. Right. The sigma subunit binds to promoter sequences in the DNA, okay? And promoter sequences typically have what are known as ta-ta sequences, lots of T's and A's, and they're about 10 base pairs before the transcription site. You can also see these referred to as ta-ta boxes, okay? And they're typically about 10 nucleotides in front of where you start transcribing, right? And why that's important, the AT sites, there are less hydrogen bonds between the 2 strands with A and T, so they're easier to separate the 2 strands. So this is really for strand separation during initiation. Right, so that's the -10 sites. There's also sites, and these are in prokaryotes specifically, sites that are about 35 nucleotides upstream that are also important. Okay, and those are sort of the up elements that the alpha subunits bind to. Right, so here is a depiction graphically of some of these different genes in E. coli and how the promoters work. So you have these up elements. We're not going to talk about any of those sequences specifically, but they can be used to express genes a little bit more than if the up element wasn't there. So genes that have that up element in front of them, you can think of those as being highly expressed genes. And again, up stands for upstream promoter. The -35 region shown here, not going to really need to know the exact sequence there. The -10 region, that's the ta-ta box. Usually have quite a bit of T's and A's in those regions, but you see there are some that have a G or a C in there as well. Okay, and then the +1 site is where the transcription starts occurring. That's where you start transcribing. So you can see that it's not just the gene, the part that's being turned into RNA that's important. There's lots of sequence before that it also comes into play here. The sigma subunit determines the types of genes that are expressed. So certain up elements, or also known as consensus sequences, are good for targeting certain sigma proteins. Okay, and so here's a list of different sigma subunits. There are multiple sigma subunits in an E. coli cell. Here's a list of them, and so they probably don't need to know any of these values in the table specifically, or even the different types of sigma subunits. Just know there are different sigma subunits, and depending on which one is used as the promoter in a given gene, tells you the sort of the function or the level of expression of that gene. So here for the sigma 70 subunit, those are general housekeeping genes, which means they're usually expressed at a pretty constant level at all times throughout the cell life. Okay, and then some other genes are shown. And why is this important? Well, you don't want to always express genes all the time, right? There's no point in expressing a gene that you don't need. So this is a way for the E. coli cell to control at what point in time a particular gene needs to be expressed. So if we look back at that problem, listing the similarities and the differences between DNA polymerase and RNA polymerase, the similarities, they both use DNA as a template, right? They're both reading 3' to 5' and synthesizing their new strands in 5' to 3' directions. Both need topoisomerases for dealing with the supercoiling from unwinding. They both need magnesium, and specifically 2 magnesiums in the active site. Both polymerases have the same mechanism. And the initiation of both starts at specific sequences. So when you're doing replication, you start at the origin of replication. When you're doing transcription, you're starting at these specific promoter regions for transcription. Okay, what are some of the differences? Well, in RNA polymerase, you need nucleotide triphosphates. In DNA replication, you need deoxynucleotide triphosphates. Okay, so that's a very slight difference. They're substrates. Proofreading, it turns out, we didn't mention this, but in RNA polymerase, proofreading really isn't as important. If you mess up a messenger RNA, that is going to have an effect when you go to translation and you turn that messenger RNA into a protein. You're going to get, a mutated protein, and it might not function very well. But that's going to be a temporary problem because your messenger RNA doesn't last forever. We'll see here in a little bit later in the lecture just how long those last, but they get degraded. and you have to continuously make a messenger RNA if you want to continually make the protein. And proteins as well, proteins have a certain lifespan, and it's usually fairly short. So if you make a bad copy or two of a protein, not a big deal, okay? It's not like in a DNA when if there's a change, it's going to be passed on to the next generation. It just makes a bad protein that then gets degraded. Okay, so you don't really need a proofreading function in RNA polymerase. Another difference, you're doing this in one direction. Your RNA polymerase is making a messenger RNA. It's only using one strand and it's going in one direction. And the DNA strands, they're not completely unwinding. They're being put back together right after the polymerase. So let's talk about the initiation and the elongation part of transcription in E. coli. Well, there's what's termed the closed complex. And that's when the DNA that's in the promoter region is bound to the polymerase, but it's not yet unwound. So you have the polymerase binding to your DNA, but the DNA is not yet unwound. That's called the closed complex. The open complex is the same thing, but now you have the promoter region in that ta-ta box, the AT-rich promoter region is separated in unwound and open. Okay. The sigma subunit will leave, right? Remember, that's the one that recognizes specific promoter regions and upstream element parts. That will leave before elongation starts. So that acts to direct that polymerase where it needs to go. The open complex will form, and then that sigma subunit will leave. So if we look at this in our picture here, our sigma subunit now is directing our polymerase. And you can think of this, we're not showing the alpha subunits and the beta and beta prime now, we're just showing it as one blob. But our sigma subunit is kind of telling it where to go. Okay, match up with this promoter region. Okay, closed complex forms. Then now the strands open in that ta-ta box. Okay, and that puts the +1 site in the first, in the kind of the active site where that transcription is going to start. Okay, so that would be binding would be the sigma directing it, the closed complex forming. Initiation would be where we're getting the open complex where the ta-ta box opens. Okay. And then before transcription can occur or elongation, our sigma subunit leaves, okay, and then elongation starts occurring. Right, and again, in elongation, we don't need any primer for this. Okay, we don't. It's not like in replication where you need a primer, an RNA primer to start. We don't need that. We just add RNA directly to that template strand, and only a small section, about 17 base pairs long, of DNA is unwound at any given time. Single-stranded RNA is stable and falls off of as a DNA helix refolds. So on this, you can think of it as kind of the lagging end, the back end of this. As that DNA comes back together, that RNA, single-stranded RNA, just comes off of that DNA. And I'm sure there's something in the shape of that polymerase that causes and facilitates that to occur. Right, this is a picture, a crystallography picture of this occurring. And here we have the polymerase subunits, and if we kind of remove one of those to see in what the DNA and the RNA look like, those are shown here. And so this is kind of a cool, more realistic picture of what's happening. And you can see here the bubble is from about -10 to about +3 in this case, right? So that would be, something like 13, 14 base pairs. That's unwound. Right, so the last step of transcription is termination. And there are two different ways this occurs. They're called row independent and row dependent. Okay, and row is just a protein complex that acts sort of like a helicase to unwind the DNA RNA complex, and it's ATP dependent. So depending on whether or not that's needed, there are two different ways termination can happen. You need a signal to stop because the RNA polymerase is very processive. So if you don't stop it in time, it's just going to keep going. So you'll go through your entire gene and then it just keeps going, which would not be beneficial, right? Because if you don't have the correct length of your gene, you're just going to be making some other protein that has no real function. Okay. This process in eukaryotes is still not that well understood. Okay, so how the first way, the row independent termination occurs in prokaryotes. The RNA polymerase will pause at certain sequence during transcription. So after your gene, you'll have a certain sequence, like a termination sequence, and that causes the polymerase to sort of pause. And then this sequence also allows the formation of a hairpin within that mRNA product. Okay, if the polymerase pauses for long enough and the hairpin forms, that RNA-DNA hybrid structure is going to be disturbed, and that promotes the dissociation from the polymerase. So a typical sequence for a row-independent termination might look something like this. You don't have to remember or memorize this sequence. But the interesting thing that occurs here is there's a palindrome in there, which can form this hairpin within the mRNA that's formed. I probably should say RNA just in general, not only for messenger RNA. Okay, so it forms this hairpin. Okay. And when it forms that hairpin there, it can sort of bind up and the polymerase kind of will stop, and then you get this termination. Right? Pauses, it turns out these pausing occurrences are pretty common in the polymerase. Not all end in termination. Okay? But if you have this where you pause, form that hairpin, it's more likely to lead to termination of the transcription. And also here, after the formation of that hairpin, you have a place where you have many A residues, right? And that's because the double hydrogen bond is less stable than a GC triple hydrogen bond. So that also helps lead to the dissociation there and the termination of transcription. The row-dependent terminations may be a little bit more complicated just because it involves an extra protein. The template here lacks the sequence of A residues, so that becomes less important than that we saw in that last slide in the row-independent termination. You still use this stem loop structure, that hairpin, that's still found in this row-dependent termination. Okay, what row is, it's a protein that binds RNA and it uses ATP. It'll move in the 5' to 3' direction, and remember that's the direction that we're synthesizing this RNA. It moves in that direction until it reaches the polymerase. Okay, once it reaches that polymerase, it sort of pulls the RNA off of the DNA because it has an RNA-DNA helicase activity. Remember, a helicase is something that unwinds double-stranded nucleic acids. In replication, we talked about a helicase, moving on each end of the replication bubble, acting to unwind that double-stranded DNA. Row is like that, but it's unwinding RNA-DNA duplex. Okay, so this is how Rho functions. Okay, it's a multi-unit, multi-subunit protein, which might look kind of similar to the helicases we saw in the pictures from last lecture. And it's moving from the 5' to the 3' direction, okay? And it's using ATP to do this. Okay? So very, very, I guess, simple picture. Hopefully this is not too hard to remember, because this is a helicase, a type of protein that we see in replication. Okay. So now when we think about a gene, like in chapter 24, we've talked about a little bit about genes, not really, more so chromosome structure. But when we think about a gene, there are different parts to them. You have promoter regions telling the RNA polymerase where to transcribe. You have the actual coding regions, which give the recipe for the amino acids in the protein that are going to be translated eventually from the messenger RNA. And you have also a termination sequence at the end that tells the RNA polymerase, okay, you can stop transcribing here. Right. Eukaryotes contain several distinct polymerases, and they involve several proteins. RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Okay, so RNA polymerase 1 synthesizes pre-ribosomal RNA, and those will be for these 28S and 18S ribosomal RNAs. We'll talk about those in the next lecture when we talk about translation. Okay, the ribosome is the place where proteins are built and synthesized. So messenger RNA gets translated at the ribosome. Okay. But those ribosomes are made out of RNA. And so RNA polymerase 1 is important for those. RNA polymerase 2 is responsible for the synthesis of messenger RNA. So probably in this class, this is the one that we'll be talking mostly about in terms of eukaryotic cells. Okay, it's very fast. It can do 500 to 1,000 nucleotides a second, very well studied. Okay, and then RNA polymerase 3 makes the transfer RNAs, okay, and some other small RNA products. Again, transfer RNAs we'll be talking about more when we talk about translation. If we look at eukaryotic RNA polymerase 2, the one responsible for making messenger RNA, it's again more complex than a prokaryotic RNA polymerase. And one of the reasons why is it needs to interact with a very complex DNA storage. Unlike prokaryotic cells, in eukaryotic cells, you have things like histones that need to be dealt with when you're transcribing the DNA. It's very large. It has 12 subunits. None of the subunits, you won't really need to know them for the exam. You really just need to know for the exam the subunits of RNA polymerase for E. coli. But we'll go through some of these. RBP1 is sort of like the beta prime subunit that binds to the DNA. RBP2 is like the beta subunit that is actually the polymerase active site. There are many transcription factors, abbreviated as TFs, and we'll see some of those here in a second. The largest subunit also has a long C-terminal tail with a peptide heptad repeat, and so that's the CDT, carboxy terminal domain. Right, so this is a list table from the book of the different subunits of some of the different proteins involved in RNA polymerase 2 in eukaryotes. Right, polymerase 2 itself has 12 subunits. You have this TBP, which binds to the TATA box. And then the other one I wanted to mention is TF2H. So TF, transcription factor, 2 standing for polymerase 2, and H meaning it has a helicase activity, right? And this is what unwinds the DNA at the promoter. Okay, and it also has 12 subunits in it. So this gets, very, very complicated. So if we look at the assembly of our RNA polymerase, assembly is going to be initiated by the interaction of the TATA binding protein with the promoter, just like in the prokaryotic cell. Helicase activity in TF2H unwinds the DNA at the promoter, and then you have a kinase activity in the TF2H that phosphorylates the polymerase, and that allows the polymerase to escape that promoter, and that's what initiates the transcription. So TF2H unwinds the DNA and then turns on the polymerase. Right, a difference between eukaryotic transcription and prokaryotic transcription is also the scale. I would say that this is kind of a difference in scale, because our TATA region, our TATA box, is now typically at the -30 position, compared to it was at -10 in prokaryotic cells. So you have just a little bit longer spacing between that TATA box and where you initiate transcription. And then you also have some various regulatory sequence still further upstream from the TATA box. Right, crystal structure here of DNA recognition within the TATA binding protein. Right, not super important for a class, but shows you how this protein recognizes that region and binds to the DNA. Right, so here we would get our TATA binding protein to initiate binding at that TATA region. Okay, TF2H comes in. There's a couple other ones that come in before this, but the one we really talked about so far has been this TF2H that comes in, creates that open complex, unwinds the DNA, and then ultimately phosphorylates the polymerase 2, activates it, and starts transcription. Right, so we get our binding first of all. TF2 opens that unwound DNA. Now it's an open complex. It phosphorylates polymerase 2. It can then detach from these other transcription factors and actually start polymerizing and making our messenger RNA. Right. That will continue, so that'll be the elongation phase, and then finally it reaches a termination sequence and stops, and our messenger RNA is released. Okay, moving on to section 2, and this is where we're going to talk about RNA processing. And this happens mostly in eukaryotes. Okay, so we've talked about transcription primarily in prokaryotes, but they're kind of at the end in that section. We mentioned how it happens in eukaryotic cells. In terms of processing, when we talk about RNA processing, we're primarily talking about eukaryotic cells. Okay. Almost all newly synthesized RNA molecules are processed to some degree in eukaryotic cells. The 5' end is capped with a methylguanosine. Introns are spliced out, and a poly A tail is built at the 3' end. So really, you can think of this as the front end, the back end, and in between. All of these pieces of the RNA are processed to some degree. Processing is catalyzed by protein-based enzymes and by RNase-based enzymes known as ribozymes. Okay, only some prokaryotes have splicing out introns, but many do process their tRNA precursors. Okay, so introns are relatively rare in prokaryotes, but their transfer RNAs do get processed to some degree. Right. So our picture here of our polymerase, and this is happening, this is polymerase 2, so we're making messenger RNA in a eukaryotic cell. Right. As this is elongating, right, we have a 5' cap to start. We have different green and yellow pieces in it. And what that represents is the green is an exon, which will ultimately end up in the mature messenger RNA. And the yellowish region are introns. Those are the ones that need to be spliced out. So it turns out as this is being synthesized, these introns are being spliced out. And what's left at the very end is going to be a mature messenger RNA that has only the exons in it. All the introns have been cut out, and it has a 5' cap and a poly A tail at the 3'. And so that poly A tail is what's referred to as a non-coding end sequence. So that's not going to be translated into protein. So first, let's talk about the capping, and that's at the 5' end. The capping protects the messenger RNA from 5' exonuclease activity. So if you have these exonucleases around that work from the 5' end, you can get degradation. Capping it here with this 7-methylguanosine, that helps it be immune to those. Okay, do you need to know this exact structure? Probably not. You just need to know that it is a methylguanosine nucleotide that's stuck to that 5' end. And remember, at that 5' end, you have that triphosphate linkage there. The capping enzymes are actually tethered to the C-terminal domain of polymerase 2. So we talked about how that C-terminal domain was, very extended and long in polymerase 2. Well, this is the significance of that. It actually has binding places, one for being able to be phosphorylated, to be turned on to start transcription, and then the other thing that it allows is a place for these capping enzymes to bind. And actually, once that messenger RNA reaches it, the 5' end there reaches that enzyme, it caps it. Okay, and so the cap is made after about 20 to 30 base pairs have been added to the messenger RNA. And then once that cap has been added, that capping enzyme leaves, and you get this cap binding enzyme that kind of binds the cap of that 5' end, and it kind of sticks it there to the C-terminal domain of polymerase 2. Right. The second form of splicing is processing is the splicing of the introns. Group 1 and 2 are self-splicing. They don't require any proteins, no high energy source like ATP, and they require only 2 transesterification steps. Group 1 requires guanine nucleoside or nucleotide cofactor, and group 2, they don't require any exogenous nucleotides. We'll look at these individually here in a second. There's a third class that isn't self-splicing. Genes in higher eukaryotes have more introns than exons, it turns out. So the processing of messenger RNA, this splicing, is actually pretty important. Right, so splicing of group 1 introns. Okay, these are found in the nucleus and mitochondria, also in chloroplasts in plants. They're in ribosomal RNA, transfer RNA, and messenger RNA. These are the ones where guanine nucleoside cofactors needed for attack. Right, and so here we have our intron, and this exogenous guanine residue is needed here to attack at a particular site, and what it does is it binds to the start of that intron, and it frees up a free end of this exon, which then can attach to the next exon. Okay, in a sense, you're just removing this yellow intronic sequence and sticking the two green pieces together. Right? RNA is a catalyst for this. All right, and just a little bit more detail how this works. We have that guanosine 3' hydroxyl, right? This very common theme here. We're just having a nucleophilic attack from that 3' hydroxyl on that free guanosine, or I should say exogenous guanosine, and it's removing that, breaking that phosphodiester bond between the last base of an exon and the first base of an intron. Okay. Right, and these are catalyzed by RNA molecules that have this catalytic activity. So they're sort of like enzymes, and we call them ribozymes. And they were discovered in the early 80s by accident by Tom Cech, and he is actually, or he was, when this was discovered, at the University of Colorado Boulder. I'm not sure if he's still there or what exactly he's doing, but for this work, he won the Nobel Prize in 1989 in chemistry, right? Which was a very short time if you think about it, only 7 years. It's a pretty short time from discovering something to winning a Nobel Prize. So this was obviously very, very important when this was discovered. And again, it was discovered by accident. How some of these ribozymes look, they can be very complicated. RNA can take some much different form than DNA can. And that's one of the reasons is because it's single-stranded, and then it can sort of fold on itself and bind with itself. And so a typical picture that you see of an RNA binding to itself is shown here. So kind of a complicated picture. Not really going to need to know this in this class. But some of these ribozymes do facilitate binding of metal ions like magnesium, which they use for catalysis. Okay. The splicing of group 2 introns, it's a similar process, but the nucleophile is a hydroxyl that's within the intronic RNA chain. So how group 1 introns, we use the guanosine residue of an exogenous ribozyme. In here, group 2 introns, we're using a hydroxyl group that's found within the intron. Okay? And it goes through the same mechanism of nucleophilic attack, and then you get this cutting of the bond, the phosphodiester bond between the last residue of this exon and the first residue of the intron, and then you can then get nucleophilic attack from that exon to the next exon, and you're just cutting out that intron. And by doing so, you're making this loop of RNA. Okay. Well, it turns out most introns are not self-splicing. They're actually the third class, and we call these spliceosomal introns. The intermediate is similar to group 2 intron splicing. They're removed by what's known as the spliceosome. And the spliceosome is made up of specialized RNA protein complexes termed small nuclear ribonucleoproteins, SNRNPs, or SNRPs. Each contains RNAs that are known as small nuclear RNAs. And this is attached to that C-terminal domain of RNA polymerase II as well, right? And so this is actually, the spliceosome is attached to that C-terminal domain, and as this messenger RNA is being propagated and synthesized, the introns can be spliced out as that's occurring. So you're not creating a long messenger RNA that then is free to flow by itself, and then it has the introns cut out of it. It's occurring as it's being synthesized. Okay. The intron has a GU and an AG at the 5' and 3' ends. So that's one way in which you can tell where the introns are. Right? And this is only in eukaryotes. Right? And so this splicing, as I mentioned, the spliceosome is on that long C-terminal domain. And so as that green messenger is being produced, right, when you come to that intron can be spliced out really as this thing is being made. This is a map of the gene of the glycine decarboxylase protein. And this is one of the proteins I study in research. And what's shown here in these blue boxes, these are the number of exons. Okay, so there are 25 exons in this gene, and in between them you have introns. And so this map, what this map is showing is all the mutations that we've found in patients that have mutations and problems in this protein. It leads to a disease called non-ketotic hyperglycemia. Right? And the boxes up here are showing where specific missense mutations are occurring that give different amino acids in the final protein product. Down here is showing where there have been larger deletions or even a duplication in the gene. But these boxes here below, kind of in between the two, are showing you where there are splice site mutations. So intronic mutations in the introns that actually cause problems in the transcript of the protein. And that's kind of a unique thing, at least for me. I was coming from not a genetic background. I didn't even know about introns. And it was just wild to me that you can have a mutation in a sequence that's not even coding for the protein, but that causes a problem in that final protein and leads to a disease. And here is another gene involved in that same protein complex. And you see that in this gene there are 9 exons and a number of introns, and you have these mutations that occur within the introns as well. Right, so the third part of processing that we'll talk about is that poly A tail that's added to the end. And there's a specific termination sequence, AAU, AAA, and that's the termination sequence in eukaryotes. Okay. Termination happens 10 to 30 base pairs downstream of that termination sequence. You get a GU-rich sequence that's beyond the termination that gets transcribed and then cleaved. An endonuclease binds these and cleaves in between. 80 to 250 A residues are added to that tail at the end, so it's quite long. Okay, that serves as a protein binding site. Right, so if we look at that in terms of our picture, right, we have our termination sequence, right, 10 to 30 base pairs downstream is where our termination occurs. We get an enzyme, an endonuclease that binds to that region and actually cleaves off this GU-rich sequence that's beyond that termination site. Okay, and then we get a poly A tail that's added by using ATPs, right? And so that's another thing, right? If you're adding 80 to 250 A's at the end of that, you're using a lot of ATP. Right, so this is a summary of many of these post-transcriptional modification events. So if we have this gene with a particular number of introns and exons, right? So the exons are all labeled in numbers and the introns are letters. We would have a primary transcript of that. Right? First we add that 5' cap. Right? Then we would need to splice out those 7 introns, those yellow regions. Those are spliced. You add some extra RNA there at the end of that poly A tail. And then that leads to what we get out as the mature messenger RNA, which then can be used to go on to translation and be translated, into protein. Right, you can get more than one gene product from a primary transcript, right? And that occurs when you get different splice sites or cleavage sites from the same primary transcript. And so an example here is just this cartoony picture where you have 2 different poly A sites. Right, and depending on where you cleave and put your polyadenylation to it, you can get 2 different mature messenger RNAs. Other examples in the book, tau proteins and antibodies are 2 examples of where this kind of occurs. Ribosomal RNA also is processed in sort of in this manner, where you have regions that are spliced out of it, and then you can get some recombination of these regions in there. It's a little bit more complicated. For this class, you don't really need to know these details, but just know that ribosomal RNA is processed as well. tRNA, that stands for transfer RNA, these are processed. tRNA, what it does is it's going to convert messenger RNA into proteins, and that occurs at the ribosome, right? And we'll be talking about that next class in some detail when we talk about translation. The 3' end of a tRNA has an amino acid stuck to it. So that needs to be added in there via processing. So a primary transcript of a tRNA looks something like this, and you get a RNase P cut at the 5' end and an RNase D cut at the 3' end. And these are done by ribonucleoproteins, so enzymes that have both RNA and protein in them. Okay. When we do that, when we make both those cuts, you have a 3' end that then you can add a particular amino acid to, right? And so you get that intermediate with a hydroxyl on the 3' end. You can get the mature tRNA with an amino acid added there. There are modified bases in tRNA, and no, you don't need to know these for this class, but here they are. There are quite a few of them. Pseudouridine is one that stands out to me, and that you could see those here in pink in this diagram. Those are all the modified bases. A 3D picture of a tRNA that's shown here. Again, where the important regions to note here, the amino acid arm, that's what's going to bind your amino acid. And the anticodon is what's going to read the particular 3 nucleotide sequence called a codon in the messenger RNA. That's going to line up with that and determine what amino acid is next in that protein. Messenger RNA doesn't last forever. Okay, the rate of synthesis of a messenger RNA versus the rate of degradation of it will tell you the steady state concentration of that, and thus the amount that is available to be translated into protein. There's ribonucleases that break down messenger RNA constantly. Some eukaryotic cells have a shortening of that poly A tail, decapping of the 5' end, and then lead to degradation. Some of the messenger RNAs are around for only a few minutes or hours. The average time is about 3 hours. Okay, so messenger RNA, mRNA, is not a particularly long-lived biomolecule. Right, there are other types of RNA. One is called microRNA, and that's abbreviated miRNA. These are pretty small, around 20, 22 nucleotides long. They control gene expression by interacting or base pairing with the processed RNA transcripts to control protein synthesis. And they were really sort of recently discovered, first in the early 90s, but then right around the turn of the century, they were actually given this name. They regulate gene expression, as we've mentioned. They bind to specific regions of messenger RNA, and they do this to alter translation. So the next step in the process is sort of what they're in a sense blocking. Right. It turns out that, or estimated, that about 1% of the human genome may encode for these microRNAs, which is actually kind of a large amount when you consider there was something like, I forget the exact number, but if you don't count the introns, about 1.5% of the genome is actually encoding proteins or genes. So 1% is a pretty large number for microRNAs. They're synthesized from larger precursors, and they're processed by two nucleases known as Drosha and Dicer. So you might have heard of these. If you go to grad school, you might run into these. Those are for processing these microRNAs. So the long precursor pre-microRNAs are made in the nucleus, then they're cleaved to about a 70 to 80 nucleotide precursor. Those are exported to the cytoplasm. Dicer cleaves these pre-microRNAs to double-stranded RNA. Whoop. The complement of the microRNA removed by a helicase, right, then that really activates it. MicroRNAs are loaded onto a protein complex such that RNA-induced silencing complex, the RISC complex. Right, RISC then binds to the target messenger RNA. How this probably looks better in a picture form, right? So our microRNA gene is here. The longer big transcripts, I should say, I think I said called this pre-microRNA. It's actually it's a step before that, is the gene product. They are cleaved first by the drosha in the nucleus to make a pre-microRNA that's exported to the cytoplasm. Dicer then cuts it further into our microRNA itself. It's a duplex, though. That duplex has to be unwound, and then the single-stranded form of that is bound to that RISC complex. That RISC complex can then target our messenger RNA, and it acts to silence it because that's bound there, and then it cannot be translated when that is bound. Okay, so this is a way of these microRNAs are a way of silencing gene expression, not on the transcriptional level, but on a translational level. And I should mention before we go on here, this basic technology is used in what's known as small interfering RNAs, which are man-made RNAs that can be introduced into a cell to do a similar, undergo a similar fate to actually bind to messenger RNA and have it either block translation on it or degrade that messenger RNA. In a sense, what it's doing is just turning off the expression of certain genes. And these have been used very, very extensively recently to study the function of particular genes. So it's a very powerful tool. Okay, the last section we'll talk about in chapter 25 is, this should be chapter 26, the RNA-dependent polymerization. And in particular, the one we're really going to focus on is reverse transcriptase. And that's the ability to make DNA from RNA. Right? We can also have a case where RNA can replicate itself, so RNA making RNA from an RNA template, and then the telomeres making DNA that are an RNA protein system. Okay, so the central dogma really started out as the information in DNA can be replicated back to DNA. That would be replication. It could be transcribed into information in RNA code, right? That's transcription. And then RNA can be turned into protein information via translation. So that's how the central dogma started. As research continued on into it, there were some other steps that have been found out, and it turns out there's examples of when RNA can be reverse transcribed back into DNA, and there's also been examples of how RNA can be replicated, can replicate itself. Right. And reverse transcription was first theorized in the 60s, but proven in the 70s by Temin in Baltimore. So this is the two that discovered reverse transcription. Right. This is in our book, and they shared the Nobel Prize in 1975 for this. And where this is, one of the places where this is really important is in viruses. It turns out viruses have this reverse transcriptase protein in them and activity, and they're termed as retroviruses. So retroviruses, a virus is usually either DNA or an RNA encapsulated in a protein shell. And in these retroviruses, they have this protein, reverse transcriptase, in them, and then they have an RNA genome. Right, as they affect a parent cell or infect a cell here, they inject their RNA into that and the reverse transcriptase protein. The inverse transcriptase will actually take that RNA sequence and convert it into DNA. Right? It converts that into DNA, and then that DNA, that viral DNA, then gets integrated into the host cell's genome. Right? That host cell, when it's incorporated in that genome, it's then transcribed by the machinery within that host cell to make messenger RNA, very similar to what was in that virus, translated that in that genome encodes for the proteins that make up that viral particle shell. Okay, so you're tricking the host machinery into making many copies of this, and you're reproducing that virus in that host cell until you've produced so many of those viruses that cell just kind of ruptures, and then it spreads to adjacent cells. Okay, so that's the way, the basic way a retrovirus works. All Right? And retroviruses typically contain 3 genes, and those genes are the GAG, which stands for Group Associated Antigen. This encodes a long polypeptide that's cleaved into 6 smaller proteins that make up the capsid. So what the virus is, the particle of the virus, so to speak. The Pol gene encodes a protease that cleaves the long polypeptide. So the GAG antigen, the Pol encodes a protease that cleaves that into the 6 smaller proteins. It also encodes reverse transcriptase and an integrase that inserts that viral DNA into the host genome. And then the ENV gene encodes the viral envelope. You can think of that as envelope. Right, so how, if we look at some retroviral genes here, these are the long terminal repeats on either end when they've been incorporated into that host genome. Right, they're going to, as transcription occurs, you get these gene products out, and then that's the primary transcript. They're translated in so that from one primary transcript, they can be translated into a number of different products, and they're cleaved into their separate pieces. Right. The important thing that we look at here in terms of this class is that enzyme reverse transcriptase. Okay, it catalyzes 3 reactions. It can catalyze RNA-dependent DNA synthesis. So that is the reverse transcription. Okay, it takes an RNA template and it makes DNA. And from that you get a DNA-RNA duplex. It then catalyzes RNA degradation. So from your RNA DNA duplex, you degrade the RNA and you're left with only DNA. And then it also can catalyze DNA-dependent DNA synthesis, much like a DNA polymerase would. So the DNA that it's left with from that initial synthesis, it can synthesize the complement strand to that. So it can make double-stranded DNA from a single strand of RNA. Right, so this new double-stranded DNA is what then can go into the host genome, which will then make the RNA protein for the new viruses being formed. This does require a primer, the reverse transcriptase, and so that's why the virus, I don't know if you noticed it in the previous slides, but the virus brings a transfer RNA from a previous host that it uses as a primer. Okay, it has no proofreading activity, so because of that, there's a very high mutation rate, and that leads to faster viral evolution. Okay, and that's one of the reasons why some viruses are very tricky, because they constantly mutate. And we've seen this, nowadays with COVID, how it's just constantly mutating and sort of always changing very quickly before you can get what's the word? Before you can get a vaccine to actually match it, it's mutating. Right. Another important use of reverse transcriptase is also to make what's known as complementary DNA or cDNA, and we talked about that in Chapter 9. That would be sort of a lab use of this enzyme. Right. Some retroviruses cause cancer. Most viruses don't actually kill the host, but they do integrate into the chromosome. If you're a virus and you kill your host, you're not going to have you're doing yourself kind of a disservice there, right? Because there's somewhat of a symbiotic relationship between virus and host. So you don't really want your host to die, at least right away. So most actually don't kill their host, but they do need them to propagate. If your virus contains what's known as an oncogene, that's something that will make the cell grow abnormally and be susceptible to turn into cancer. So the example here is the Rose sarcoma virus has the SRC gene, and that's encodes a non-receptor tyrosine kinase that affects cell division. Right? So this gene in that Rose sarcoma virus will affect the cell division in the cells that it infects. HIV is another example of a retrovirus. It invades and kills T lymphocytes. The HIV reverse transcriptase is very, very error prone, right? More so than a typical reverse transcriptase. And because of that, that's one of the reasons why it's very difficult to create a vaccine for HIV, is because that transcriptase, the reverse transcriptase, is so error prone that it's just constantly making mutations, and any vaccine you make is not going to be effective because the virus has, the outside of the virus has changed. So that leads to the drug targets for HIV, the actual protease that cleaves the gene, the primary transcript, into the multiple subunits, and also targeting that reverse transcriptase itself. And here are just an example of some HIV drugs that target the protease and the reverse transcriptase. You might have heard of these, the AZT and the DDI. And I should mention that, right, they lack that 3' OH group, so the lack of that 3' OH group stops reverse transcription. There's some non-retrovirus use of RNA replicase. The 5' to 3' synthesis of RNA from an RNA template. So this is analogous to a DNA replication, but you're replicating RNA. And specifically, the RNA genome of viruses or phages can be replicated in this manner. These will not work with DNA. The RNA replicases, unlike reverse transcriptase, RNA replicase do not work with DNA. The last thing we'll talk about are some well-characterized ribozymes. Okay? We talked about self-splicing group 1 introns. That would be an example of a ribozyme. The RNase P and RNase D that cleaves those precursors to transfer RNAs, those are ribozymes. There's one called the hammerhead ribozyme. This cleaves RNA of virusoids. So there's circular RNAs that are replicated by plant viruses. It's so named because it looks like the head of a hammer. Okay? But this is involved in plant biology, so we really don't cover that a whole lot. The others are telomerase. It's an RNA that works with protein, and then the ribosome itself, which we'll be covering next chapter, and that's an RNA that works with protein as well for translation. Right, so here we have a ribozyme, just a basic picture of a ribozyme, the hammerhead ribozyme. It looks kind of like a hammerhead. It's very small. Right, so in terms of telomeres, we mentioned telomeres in chapter 24. These are the ends of the chromosomes. They have tandem repeats, and it's this repeating TTA, GGG, repeating over and over again. They can be tens of thousands of base pairs long in mammals, so they can be really, really long, and they tend to get shorter as we age. Right? And some people think that this is really the aging process has a lot to do with our telomeres shortening. this ribozyme telomerase was discovered by Carol Greider and Elizabeth Blackburn. I actually got to hear Elizabeth Blackburn give a talk about this, which is really cool. They won the Nobel Prize for this in 2009, and so pretty recent. It was just, they were discovered in 1984, so that kind of shows you that quite a long period of time, right? 25 years from the discovery to when they won the Nobel Prize for this. Right? What these are is it's a mixture of RNA and protein. And so our RNA is shown here in the center, and the protein, mostly an alpha helix, is shown around it. Okay? The RNA is involved in base pairing. These help rebuild the sticky ends with no template. So RNA in the telomerase provides that template. The problem with the linear DNA, that, like we have in our chromosomes, is with the ends. So if you want to visualize this problem that occurs when trying to replicate the ends of chromosome, this is a good video to do that. Okay, so what telomerase is doing, it's using the overhanging ends on a linear piece of DNA, and the RNA part of the telomerase is binding with that template. Right, so you have this RNA in your telomerase enzyme, that's your internal template. Right, so these sticky ends, they're not being copied or repeated by your DNA polymerase. Okay, and so that's where these telomerase enzymes come in. All right. It's using a type of reverse transcriptase. So RNA is used as a template to make DNA, which is the DNA specifically in your telomeres. All right, you're going in the 5' to 3' direction. That's the usual direction of synthesis. Okay, once removed, those TG sticky ends of DNA remain. And the DNA polymerase can then fill those in. Here's just another figure of kind of showing how this is happening. So your telomerase, in a sense, can help elongate your telomeres when they're shortened by cell division. So in summary, the RNA functions that we've talked about, primarily we're talking about messenger RNA, and that's RNA that's encoding, going to encode amino acid sequences of the polypeptides found in the cell. So that's involved in translation. Transfer RNAs, those are the ones that are also involved in translation, but they match specific amino acids in triplet codons. So they're involved in translation as well, but that's where the amino acids come into play. And then finally, ribosomal RNAs, those are the constituents and the catalytic components of the ribosomes, which again will be used in translation. So all of these RNA functions are going to, we're going to talk about them again next lecture when we talk about translation. Ribonucleic acids also play several other functions in eukaryotic cells that are less well understood. MicroRNA appears to regulate the expressions of genes. Ribonucleic acids also act as genomic material in viruses. And then there are ribozymes that are catalyze reactions. So they're RNA that helps catalyze reactions, and specifically those self-splicing introns we talked about and the telomerase. But the ribozyme will also be in that category, but we'll talk about that in chapter 27. And because of this, The discovery that RNA can actually catalyze reactions has really narrowed down there was always sort of a debate, what is the first biomolecule? Was it amino acids? Was it DNA? But because RNA can actually catalyze a reaction like a protein or an enzyme can do, and it can also hold information like DNA can do, it's really sort of leads it to kind of narrows it down that it was probably the first biomolecule that arose and it allowed life to evolve. And when you do simple experiments like taking things like cyanide or ammonium cyanide in reflux them, very simple chemical compounds that were on a primitive planet, and all you do is heat them up and cool it down in a reflux apparatus, you get out these the same kind of compounds that you see in the bases of nucleic acids. So these compounds are not that hard to synthesize and make. So it really kind of cool if you're into that sort of evolution or thinking about these unanswerable questions. Really, where did life really start? It looks like it probably started as an RNA molecule.